Muhammad Umar Farooq biological video. Please make sure to subscribe, like and share. Ask the question for answer and comment about the artwork. You can also answer question to each other for solving the problem in comment below. So today we will discuss about the cholesterol and lipoprotein metabolism and their transport of the cholesterol and lipoprotein, which that is fat. So important to know that here is the cholesterol is a part of plasma membrane, you know, and produce a hormone, a different type of hormone, and steroid, which that is the precursor of the cholesterol. And precursor of the hormone and steroids, that's cholesterol transported in the body cell through lipoprotein. So the lipoprotein is a globule-like structure which contain apoprotein, phospholipid, triglycerides, and as well as fatty acid. So let's begin the understanding and just focusing on the cholesterol for getting the concept. So here is the digestive system of the human being, so which contain a digestive system. When we will eat the fat, which that is go into the stomach, that is known as the bolus, and after the going bolus into the duodenum is known as chyme. So here is the gallbladder, which then is contain a bile duct. So that is the gallbladder, which contain a bile acid and bile pigment and cholic acid, that is the and bile salt, which that is used for the emulsification of fat. Emulsification mean the modification of micelles. So let's talk about the understanding of the absorption of fat. So here I am drawing the enterocyte, is also known as brush border cell, which contain brush border enzyme. But here we will discuss about the fat, which that is digested from the pancreatic lipase enzyme, which release as a exocrinely, and gallbladder secrete the bile pigment from the bile duct, which that is ultimately attached with the pancreas to excrete both. So here is the CCK enzyme, which that is contract the gallbladder to excrete, which that is the stimulus of the fat. When fat enter into the duodenum, that stimulation of the bile contraction will be occur by the CCK. So here is the pancreatic amylase enzyme and pancreatic lipase enzyme. So here we will discuss about the pancreatic lipase enzyme and not amylase, and bile acids, bile salts, etc., which that is produced, and that production is create the emulsification. So the emulsification of fat, which convert into the micelle. So the micelle structure is look like this, which can be amphiphilic or monophilic. For example, the hydrophilic side is two, for example, inside and outside. So that is known as the amphiphilic. Why this is important? That is why, because it's is attached with the bile acid, etc. When enter into the enterocyte, so the amphiphilic micelle will be occur. So when go inside of the enterocytes, in the enterocyte there are the endoplasmic reticulum, which give the protein, and Golgi bodies will modify it. So the modification by the ApoB48, which that is produced, which that create the chylomicron. This chylomicron contain very low density lipoprotein. That is the initial phase of the production of lipoprotein. Ultimately, that's go into the lymphatic system. Is the lacteal is here green color. When go inside of the lacteal, that's ultimately first that is checkpoint for it to microbes are present or not present, for the immune cell activation to check it. So after the moving lymphatic system to inside of the blood vessel, and that's contain very low density lipoprotein, which this very low density lipoprotein is known as a chylomicron or chylomicron. When enter inside, here is the lipoprotein lipase enzyme are present in the endothelial cell of the blood vessel. And these endothelial cell of the blood vessel contain a lipoprotein lipase enzyme, which that used to break down of the fat and cholesterol, which that's enter into the tissue. After the entering, that can be cholesterol, fats, protein. And when fats, cholesterol, protein will be enter into the cell, that's after all convert of the ideal. So ideal, you can say that's very low density lipoprotein is also. So that is the chylomicron is remnant. Remnant means the left behind. That's things is known as the remnant, and that's VLDL remnant will go into the liver from the remnant chylomicron receptor, which that receive the ApoE site, apoprotein E site. So the ApoE is it means that there are many apoprotein which that is come from the LDL uh, LDL low density lipoprotein, which give the ApoE site. So this modification is the ApoE side to give a modification of the ApoE to receive it. So here is the thyroid releasing factor, which that's come from the hypothalamus to go into the pituitary gland of the anterior lobe to produce thyroid stimulating hormone, and these thyroid stimulating hormones stimulate the triiodothyronine and thyroxine, which that activate the receptor in the liver. So that's the mechanism. And here is the remnant chylomicron receptor, which that receive the very low density lipoprotein. So important to know that the remnant chylomicron will enter, and that's entry by the endocytosis. So the endocytosed particle is known as the endosome. Endosome contain also receptor, but receptor will be separated, and the chylomicron will be separate, very low density lipoprotein. After all, that is the lysosome will bind it to produce the cholesterol, to bind to lysis, and that's breakdown of this particle to produce cholesterol, fat, and that is ultimately either go into the bile to excrete excess cholesterol, or go into the basically high density lipoprotein empty. Cholesterol is synthesized into the liver from the glucose as well as other fats. So next diagram we will discuss in this video about the synthesis of cholesterol a little bit. So first of all, we should understand about the mechanism for absorption and releasing. So the excess cholesterol will be excreted through the bile. On the other hand, here is the fat, which that again convert into the high density lipoprotein empty, which is empty mean which that receive the triglyceride from the tissue and fatty acid, fatty acid and phospholipids. So here is the blood vessel, which contain LDL basically, and that's LDL is come from the liver. So it means the LDL mean low density lipoprotein. So the low density lipoprotein is the basically contain the receptor protein is ApoB, ApoE, and ApoC2. So the ApoC2 is used for the lipoprotein lipase enzyme to give the storage into the adipocytes, so in the form of triglycerides, and again that is conversion. So here is the very low density lipoprotein give the ApoE receptor to the low density lipoprotein, which they receive low density lipoprotein, and that's low density lipoprotein convert and receive the ApoE site. So the ApoE site is important for the basically ApoC site is important for the adipocyte adipocyte receiving mechanism for the receptor. On the other hand, ApoC2 is used for the ApoC is used for the other tissue to donate the phospholipids and cholesterol and triglycerides. So the triacylglycerol will be provided from that. On the other hand, here is the triglyceride, which that's ApoE used to. Here is the also ApoE used to give the cholesterol and fatty acid triglyceride to the adipose tissue. On the other hand, here is the hormone produced from the adrenal cortex, and that's the adrenal gland will produce hormone by the LDL receptor to receive the cholesterol, as well as reproductive organ is also as well. More and more, we will further discuss about the plasma membrane will receive the cholesterol. So first of all, we should understand about the VLDL, IDL, and LDL as well as HDL. So here is the VLDL, IDL, LDL, and HDL is further go. So here is the direction. It means the VLDL will decrease into the IDL after donating the triglycerides, and that's slope is look like this. Cholesterol will be decreased from HDL to VLDL. It means the VLDL contain less amount of cholesterol, so the HDL contain high amount of cholesterol for donating to the tissue. So HDL is come from the liver and VLDL come from the diet. So the triglyceride is maximum into the VLDL. On the other hand, IDL is decrease, LDL more decrease, and HDL is the very minimum amount of triglyceride. And ApoE site, which that is the receptor site, is the apoprotein, which that is used and increase from the VLDL side to the HDL side. Why? Because it donate the cholesterol to the ApoE site. So the donation is decreased when the cholesterol will be cholesterol will be received. So it means the HDL, which contain a high amount of cholesterol. So the cholesterol absorption from the tissue is through the ApoE. So the ApoE will be decreased. On the other hand, ApoB will be increased when cholesterol will be increased. Why? Because ApoB is provide the cholesterol to the other membrane, cell membrane, as well as going to the liver. So the liver contain a ApoB site receptor. So here is the general structure of the cholesterol. Chylomicron or lipoprotein, high density lipoprotein, contain cholesterol ester as well as the cholesterol. Cholesterol ester mean which that is ester esterification to bind with the membrane like structure is the phospholipid attachment. Look like that's plasma membrane. On the other hand, here is the ApoA site, ApoB site, ApoE site, and that's ApoC2, which that is a different type of receptor like structure of the protein. It is also known as receptor like protein. On the other hand, that was the low density lipoprotein contain just only ApoB. So the ApoB mean low density lipoprotein contain 100 ApoB. So the ApoB mean which that is contain high amount of cholesterol but the low amount of triglyceride when after the donation to the tissue. So the LDL low amount of triglyceride, 10% and more ester of cholesterol. So the cholesterol is more. It means cholesterol is absorbed to convert the high density lipoprotein that is going on toward the high density lipoprotein. So the LDL contain high number of cholesterol. On the other hand, the VLDL contain a low number of cholesterol because it's used for the absorption. So here is the basically adipose tissue which that's bind with the ApoC2 site to donate the triglyceride. That is known as the IDL. IDL is filled with the cholesterol and triglyceride is still decreased 20%. Why? Because it's VLDL convert into the IDL, and that's VLDL contain a high number of triglycerides, but IDL contain a less number of triglycerides. So the further going on. So here is the IDL filled with the cholesterol, but triglyceride is not maximum, is 20%. You can say that. So the lipoprotein lipase enzyme used to transport the triglyceride into the adipose tissue. So here you can watch this diagram to understand. On the other hand, here is when go inside the triglyceride. Sometime adipose tissue will donate the triglyceride to the IDL, and IDL convert into the VLDL, which that is very low density lipoprotein, and that is known as the triglycerides. So that mechanism is very complicated to understand, but we should try to understand as well. So here is a VLDL, very low density lipoprotein contained here, that is the triglyceride is more, and it means the more VLDL mean the ApoE site will be created. So the ApoE site will be more, and that's bind with the adipose tissue, and so the triglyceride is a 50% and donate to the adipose tissue. And after the donation of VLDL, which that is converted into the IDL. So here I am drawing that is the LDL. So IDL, a VLDL convert into the LDL or IDL. So first of all IDL and after LDL, because it's losing their triglycerides. So the triglyceride is the fat which that is the strong deposition into the adipose tissue in the form of fat. So that's important to know that that is the storage property of triglycerides. Here is the liver, which that's contain a blood vessel. It is known as the adipocyte. So we will discuss about the here is first of all that is the high density lipoprotein. High density lipoprotein mean the ApoB is more and cholesterol is also more. So the ApoB side is used for it to absorb the cholesterol. Why? Because high density HDL empty, if we will talk about the HDL empty mean triglyceride is empty, so the cholesterol is more. So HDL is empty, so the cholesterol will becoming more. Why? We will discuss about here. So here is the donation of cholesterol is here, and that is, if we will talk about the absorption of cholesterol, that is the esterification, and that's esterification is the oxidizing of the cholesterol which that's bind with the surface of the high density lipoprotein empty to become a high density lipoprotein filled. Filled means the high number of cholesterol are present inside of the HDL, and it's donate to another plasma membrane where the need where the need of cholesterol, triglycerides, etc., which that provide to the plasma membrane for normal function. On the other hand, remnant HDL field is go into the liver, which contain a high number of cholesterol which that absorb from the plasma membrane as well as other from the blood, which that go inside and excrete through the bile, and that is go into the liver. So I hope you make sense about here. Here is the HDL field, and here is the plasma membrane which that is absorb, either not absorb and go into the filled HDL into the liver, which that is using further or excreting excess cholesterol. So important to know that here is I am drawing the structure of liver which that's contain hepatocyte. Hepatocyte mean the liver cell which carry the glucose through the glut transport 2 channel, which that is sugar will go inside to produce glycolysis to produce pyruvate, and pyruvate go into the mitochondria to produce acetyl CoA, and this acetyl CoA will either go into the tricarboxylic acid cycle to produce ATP or go outside, is known as the uh, efflux of the acetyl-CoA. So that's high HMG-CoA reductase enzyme used to it to produce malonyl-CoA. On the other hand, that is the statin is the drug which that is inhibit the HMG-CoA reductase enzyme to inhibit the malonyl-CoA. Anyhow, malonyl-CoA convert into the cholesterol, fatty acid and triglyceride from the monoglyceride and diglycerides. After all, these will be cholesterol is go into the it's if excess of cholesterol will store into the bile and excrete into the digestive system. On the other hand, if we will talk about the new synthesis of cholesterol will be developed, and that is the lipoprotein production. And here is the DNA, which that is the transcription to translation to produce messenger RNA, and that's go into the produce apoprotein, and this apoprotein is bind with the cholesterol making systems, which that is the lipoprotein. And this lipoprotein is three type: high density lipoprotein empty mean the less amount of cholesterol, and high density of lipoprotein filled is mean the excess of cholesterol are present. On the other hand, the LDL, which that is the low density lipoprotein. So the low density lipoprotein contain a high number of triglycerides. So these three lipoprotein which provide the different mechanism, which provide the nutrients or substance from the different mechanisms. So here is the transport triglyceride and cholesterol as well as fats from the high density lipoprotein field. So the basically cholesterol transportation through the HDL field. On the other hand, LDL, which that is used to provide the fats to adipose tissue. So it's providing the adipose tissue to the LDL from the LDL receptor of the tissue. On the other hand, that is the here is the HDL empty, which that's pick the cholesterol. So one is for picking, one is for providing, and another LDL is used for the provide the fats to adipose triglycerides. So here is when go into the blood vessel, which HDL field contain ApoC2. On the other hand, HDL empty contain a less amount of cholesterol but excess of other. So ApoC2 is attached with the endothelial cell to provide cholesterol, and HDL empty, which that's pick the cholesterol, and ApoE site, ApoB site are present. On the other hand, the LDL, which contain only ApoB100. So what is mean? Which that is attached by the B site. So here is the adipose tissue, which that is lipoprotein lipase enzyme to add it. So important to know that this is in the form of cycle. For example, the HDL, which that is filled, is go back into the liver, and again the cholesterol, excess cholesterol, either store into the bile or use again. On the other hand, the HDL empty, which that is first of all pick, and HDL filled, which that is provide the cholesterol to the tissue, but if not required, so go back again into the liver for bile excretion. So important to know that that cycle is created, which that is the lipid produced from the liver, which that's provide the tissue, and cholesterol provide the tissue, either use as a cholesterol in the form of hormone production, or if excess in the body, which that's go back to create, and that's give the signal to go back into the liver for bile excretion. So that is all thing is done. So the very low density lipoprotein chylomicron, which convert into the remnant after the using to go into the liver as a low density lipoprotein. But on the other hand, that high density lipoprotein is produced from the liver. So I hope you make sense about that video. Please comment below to ask the question for answer.